So in photosynthesis, yesterday I gave a explanation for dark reaction, and some of you asked about light reaction. So as I told, photosynthesis, the physiology is divided into two parts. One is light reaction and other one is dark reaction. In dark reaction only, we can able to find two types. In plants like rice, wheat, remember these are all very important example, potato, bean, and two special examples, tomato and bell pepper. All these plants are called as C3 plants who do photosynthesis in mesophyll cell only. Whereas in plants like wheat, maize, sugarcane, amaranthus, sorghum, the process of dark reaction is called as C4. Remember, C4 has C3 also. Why I am saying like that? Because the process of photosynthesis, dark reaction, occurs in two cells. Initially in mesophyll cell, followed by bundle sheet cell. Can any of you tell what kind of reaction in mesophyll and what kind of reaction in bundle sheet? You can text in comment box. Very important topic, very easy topic. So yesterday I explained about only C3 cycle. And now I'll explain about light reaction first. Then I'll go to C4 also. You can even remember C3 plant. If carbon dioxide is less, then it can switch over to photo respiratory pathway, which can also be called as a C2 process, C2 cycle. C4 plants are always C4 plant whose photorespiration is very, very less. So to your level, you can say no photorespiration in C4, whereas C3 plant has C2 cycle also. C4 is always C4. You keep in mind like that. Repeat the example. Dark reaction. Plants like rice, wheat, potato, bean, tomato, and bell pepper. C4 examples are maize, sugarcane, amaranthus, sorghum. You remember any one example strongly. Better to remember C3 examples. I used to say, remember it like common plant which we consume daily. Rice we are eating daily. Wheat, you remember like that we are eating daily. Say for example, we consume wheat daily. Potato, bean, tomato, bell pepper. Just for a memory I'm saying, you keep one thing in your mind strong, other one you can eliminate this and answer. And C3 plant do C2 photorespiration, very important. Now if I take light reaction occurs in stromal lamellase. Light reaction occurs in mesophyll cell, chloroplast. In chloroplast, we have a membrane-bound structure called thylakoid. So if we take a mesophyll cell, it is an elongated cell. Hope you remember what is palisade parenchyma, which is elongated cells, which has a nucleus and which has a chloroplast. From the lesson cell, remember chloroplast is a double membrane-bound structure. It has its own DNA, ribosome of 70S kind and amino acid. I already given a lecture about a lesson cell. Kindly follow that. Now, if I take this chloroplast out and enlarge it, we can see it is a disc-shaped organ. As I already told, it is a double membrane bone structure which has a liquid part. The liquid part is called as stroma, and it has a membrane bone structure which is called as a thylakoid. Imagine little 3D manner, thylakoid. If many thylakoids are arranged or piled, stacked, one above the other, then we say we are getting a structure called grana. There are many such granas are there. One grana to other, there is a lamellae which runs in between two granas. This lamellae, since it runs in stroma, we can also call it as stromal lamellae. Stromal lamellae has only PS1, whereas thylakoid has both PS1 and PS2. PS1 and PS2. If PS1 alone present, then cyclic photophosphorylation will occur. If PS1 and PS2 both are present, then most favorable is non-cyclic. Since PS1 is there, right, it can even do cyclic photophosphorylation. To explain that, I'll take one thylakoid out from here and we'll enlarge it further. If I take only one thylakoid and enlarge it, I have to draw a structure like this. One thylakoid is a membrane bone structure. You remember structures like this. It has membrane on all sides. I used to say, you can imagine your pencil pouch or a pencil box. A pencil box has a plastic on all sides, top, bottom, right, left, everywhere. And inside there's a cavity where you keep your materials. Like that, the cavity is called as lumen. The material, the flat material, nothing but membrane, where lot of pigments are there. For our easiness, let us take only two regions. Let me mark like one region is having chlorophyll A, other region except chlorophyll A, that means chlorophyll B, carotenoids and xanthophyll, which is called as light harvesting center. Chlorophyll A is called reaction center. Chlorophyll A will be activated under 700 nanometer of light. So this is also called as photosystem 1. So join both this word, we can shortly call it as a PS1, sorry, P700. Same way, photosystem 2, which activate under 680 nanometer, we can jointly call as a P680. So let me erase all the content so that I can explain about the light reaction alone. So what else you have to remember here? Remember, lumen has water. I will erase that word lumen also. Lumen has water. For a calculation, let us have 2 molecules of water. Chlorophyll A has hydrogen. Let me take 2 hydrogen. This is P700, right? This is P680, right? Which does not have anything. No hydrogen, no electron, no proton. Whereas outside is stroma. Stroma has high amount of hydrogen proton. Very simple. Point number 1, lumen has water. Point number 2, chlorophyll A has hydrogen. Point number 3, stroma has hydrogen proton. Mark it. Now, because of sunlight, let me bring a sun here. From sun's light, when sunlight falls on the water molecule, this water molecule is broken. If 2H2O broken, then what we get? We get 2H2 and O2. So this is the reason, or we always say, right, plant produces oxygen during photosynthesis. So this is what the oxygen is. This oxygen will go out to the atmosphere itself. It totally goes out. It goes to the atmosphere. So I'll erase that hydrogen now. Sorry, that oxygen now. Hope I can erase these lines also. So what is remaining? 2H2 is remaining. This 2H2 will be taken by accessory pigments called chlorophyll B, carotenoid and xanthophyll. So as I told earlier, there is no hydrogen at photosystem 2. Now it got hydrogen because of photolysis of water. Photolysis of water. We can also call it as photosplitting of water. Photosplitting. So because of photosplitting, which occurs at the inner side of the thylakoid. Let me write it again for your easiness. 2H2O, 2H2 and O2. O2 went out. 2H2 taken to the photosystem 2. Now, again, because of sunlight of 680 nanometer and 700 nanometer in photosystem 2 and photosystem 1. So 680 nanometer is little less, 700 nanometer is little high. So because of these two light energy, the hydrogen present in both the places now will be broken. So what I should get, if I break hydrogen, we should get, that is 2 molecules of hydrogen are broken. So we should get 4 electron and 4 protons. Electrons are excitable. As soon as electrons were produced, let me erase the hydrogen and write electrons and protons were produced. Electrons and protons were produced at photosystem 2. Since 700 nanometer of energy is given, this electron can go away up to where? Since high energy of light is given, electron can goes to the stroma itself. You mark its path. Shall I mark like this? Electron totally went out. If electron goes out, it can join with the proton. As I already mentioned, more amount of proton is there in stroma. Since 4 electron goes out per reaction, let me take 4 proton also. If both of them join, can I say once again 2 hydrogen will form? Whenever hydrogen forms, there is a hydrogen carrier. A hydrogen carrier called 2 molecules of NADP will come and pick the hydrogen, take the hydrogen, and it became 2 molecules of NADP H2. For this enzyme, NADP reductase. NADP reductase is helping here. So because of NADP reductase and NADP, as soon as hydrogen forms, it has been taken by NADP, so it became NADPH2. So this is what the very first output of light reaction. We got two molecules of NADPH2. Listen, as I told, from photosystem 1, electrons excited and went to stroma and became hydrogen picked by NADP. What about proton? Protons are remaining there only. Can I say then for photosystem 1, electron deficiency has happened as it has gone out? We can say electron deficiency or electron hole or electron lacking. To compensate that electron lacking, even at photosystem 2, electrons are formed, right? Little less energy, 680, right? This electron also excite, but since energy is little less, it can go up, excite to certain level. Later, there is an electron carrier. I'll write outside first. Electron carrier called plastoquinone, followed by cytochrome B6, followed by cytochrome F, followed by plastocyanin. 4 electron carriers are there. Let me mention first plastoquinone, second cytochrome B6, third cytochrome F, and fourth plastocyanin. In this order, that electron will be transported to photosystem 1. I told electrons are lacking at photosystem 1, right? So for that lacking of electron, this electron will go and balance it. So as electron comes to photosystem 1, it can join with the proton already available. Can I say now, if both join, once again, 2 hydrogen will form. I can mark the 2 hydrogen once again produced. And it is ready for next reaction. Let us trace the path. Can I mark it like this? Now observe only the green dotted line. It resembles which alphabet? Give in the comment section. It resembles the alphabet Z. Correct. So we call the electron transport. Let me mark all the electron here. Plastoquinone has taken it. Cytochrome B6 has taken it. Carried it. Cytochrome F, plastocyanin, and the electrons are finally deposited or given to photosystem 1. So now just trace the path as I told. We are getting alphabet called Z. So electron transport system is also called as Z scheme reaction. It is cyclic or non-cyclic? Observe the electron. From photosystem 2, it has came to photosystem 1. From photosystem 1, it has gone to stroma. The electron has not returned anywhere. That means it is not cyclic. It is a non-cyclic pathway. Sometimes it can also be cyclic. For that condition, only PS1 should alone be active. That I am not explaining now. I don't want to confuse it here. You remember what we are discussing now is only non-cyclic photophosphorylation, non-cyclic reaction, electron transport system. Hope you understand. Now answer my question. Whenever electron goes to stroma, it has taken proton from stroma. Then can we say slowly in stroma, if this reaction continues, the amount of proton will be less going down. And see what happened to this proton which is in photosystem 2. This proton will enter to the lumen. So if this reaction occurs continuously, Remember, amount of proton at stroma will be decreasing. Amount of proton at the lumen will be increasing. Hope I understand. This is clear. So amount of proton is decreasing at stroma. Amount of proton is increasing at the lumen. I understand what is your doubt now. I am entering into the content called chemiosmosis. Chemiosmosis is nothing but transportation of proton from higher region to lower region, from lumen to stroma. What is the condition for that? First condition, there should be a proton gradient. Proton gradient. It is there, high proton in lumen, low proton in stroma. Second condition, we require a membrane. Of course, membrane of thylakoid is there. Third condition, we require a proton pump, which is called as F0 and F1 molecule. This is a protein. I'll show it where it is. Then fourth condition, enzyme called ATP synthase. ATP synthase, ATP synthetase, ATPase, all mean the same. Now, this is the condition for chemiosmosis. I'll keep it